right. We are live. So all classes are back to 100% face-to-face, right? Still going to keep live streaming, so it gets recorded and goes on YouTube. Still do the attendance quiz, so as far as I'm concerned, nothing's really going to change in this course. Even with the labs, if we're doing something in the lab where we have an introductory lecture or something that goes through how to do calculations, we'll probably also have that recorded and get it posted. So if you happen to be in quarantine or isolation, you're not going to miss anything. You shouldn't miss anything. All right. So this week we have lab. Lab's a bunch of calculations to learn how to do biodiversity measures. All right. We're going to use Excel. We're going to show you how to do it in Excel. You can do it by hand. It's just Excel's faster. We'll make use of the formulas and the fill function, fill down rows or fill down columns and so forth. So we can use the computers that are in the lab. If you have a laptop or if you have a tablet and want to learn how to do it on those, you can. I know there's ways to use Google Sheets to do these calculations. So if that's what you have, that's what you have. I'll be able to help you out and figure out how to do it, how to use it. All right, so bring that. As far as how long do I think the lab will take this week, just kind of depends on how quickly you work. So I can tell you if we do it by hand, we're in there most of the lab time. All right, but if we use Excel, it gets calculated pretty quickly. And usually, I think last year we were done by 4. But again, it just kind of depends on how quickly we do the practice problems. Next week, we'll continue biodiversity. We'll actually compare measures, like visualize. What do these numbers mean? What do the communities look like? And then the week after that will be an outdoor lab. So we will go out to the Red Arroyo. If you can drive and take some people, that would be great. If you can't, then hopefully we have enough people that are driving to catch rides. All right, it's here in town. It's right at the Saul Ross parking lot at the Red Arroyo. We'll go out. We'll mark grasshoppers and do a mark recapture study. This one is all of the labs work together. So we've got initial marking on Monday, then we've got recaptures on Tuesday, Wednesday, and Thursday to estimate our populations. We'll go out rain or shine. So if it happens to be raining, we're still going out. Those grasshoppers are still out there. They'll stink, but we're still out there. All right, and the last 2 things, quizzes. All right, we've got our last quiz that is still open until tonight. We have our Hardy-Weinberg stuff, unlimited attempts. I haven't taken the highest grade, so if you did it last week and you want to go in and retake it just to confirm you know how to do this in time for the exam, today and tomorrow it will be your last chance to do that. All right, and then I'll go through and look at the answers and correct any of the roundings, anything that was marked wrong because of rounding, because it wasn't exact is one of the answers that I listed. All of the quiz keys go live on Wednesday. Probably, I think maybe at 12:30 in the morning, something is when they go live. But that means that if you didn't take any of these quizzes, if you missed the quiz, this is your shot to retake it at a late penalty. And then lastly, our exams on Friday. So the exam is true, false, multiple answer for most of it. And then we have definitions, which I give you a word bank, give you the definition, you write the word on the blank, and then some Hardy-Weinberg calculations. Equations will be included on the exam. All right, you've got 50 minutes. We'll come in. As soon as I get here, I'll pass out exams, and then you have until 8:50. We do have a class after us, so at 8:50, I need you to finish. You will need a calculator for the Hardy-Weinberg stuff, so make sure you have a calculator. All right, and it can't be a cell phone calculator. It needs to be a calculator. All right. Any questions? Start reviewing. Yep.
Is this week's lab on our test?
Nope. start reviewing. All right, start looking over your notes. If you have questions about the notes, bring them to class on Wednesday. We can answer them. We don't do a review session at all, so if you've got questions, ask on Wednesday, and then you can always email me. Just know that you don't wait until Thursday night to email, because I might be in bed when you email me. Right? You might not get a response. All right. Moving on. So this is all new material. This is all exam 2. All right. So we finished up with thermal adaptations. What we're going to do now is talk about water adaptations. Water is both a resource and a factor, physical factor. All right. So organisms need water to assimilate. They also need to be in such an environment where they can not dry out. It has to be moist enough to where they don't dry out. So what we're going to do is we're going to split this into animal adaptations and plant adaptations. All right? So in order to understand water, the problem of water, we have to understand how water moves. Now, this should be review. This really should be review. I remember learning this in at least in middle school. I don't really remember what all was covered in elementary school, but at least in middle school and then in high school and then in college in both biology and chemistry classes, all right? And it's osmosis. All right? So osmosis is the movement of water through a semi-permeable membrane. And that's what we're interested in. How does it move? What determines how water is going to move? All right? Well, it's going to move based on its water potential. And the water potential is the energy gradient between our two systems caused by the relative water and the solute concentrations. All right? Water is going to move from a high water potential area to a low water potential area. So same thing, just follows its gradient. All right, so it follows its gradient. But what determines the gradient? Well, the water gradient is determined by these 3 different things: the osmotic pressure, or the osmotic potential, the pressure potential, and the matric potential. All right? These 3 things determine the osmotic or the water potential, and that tells us which way water is going to flow. Oops. Fix that. Misspelling. All right. So, there we go. Determine water. All right. So, our osmotic potential is the energy due to the solutes themselves. So, the salts, the sugars, your metabolites. Right? And in our concentration, water is going to move from its high potential area to its low potential area. But when we think about salts and sugars and stuff, water is going to move from a low salt area to a high salt area. And what it's trying to do is try to get the concentrations on both sides of that membrane to be equal. All right, so in this image, we have a high salt concentration and a low salt concentration. Right? In terms of the osmotic potential, this has a higher osmotic potential than this. And thus our water potential is going to be higher on this side than this side. So if water can move through this membrane, it's going to move towards the right to try to equalize our potentials, make the potential 0. All right, so it goes from high water to low water, or flip side, it's from a low salt to a high salt solution. That's how the osmotic potential works. The pressure potential is that water energy that's due to the pressure exerted by muscles, cell walls, and any other mechanism. All right, so with our osmotic potential, we're not looking at any pressure that's happening in the system. But if we put it inside a cell, right? The cell has a membrane. And the membrane can swell and it can shrink. Well, when it swells, much like a balloon, it's going to exert pressure on all of the contents inside. And that's going to affect our water potential. In plant cells, we refer to this pressure as turgor pressure. All right, so in a plant cells, they have the cell walls. They're going to be somewhat rigid. And that force of the cell walls on the interior of the cell is going to increase the pressure. That pressure can affect how water can move in, because if water's moving into a system and it hits a pressure that's too high, it's going to stop moving, even though the salt concentration says you need water, you need to keep coming into the cell. The pressure is going to counteract that. It's like a balloon. The more air you put into the balloon, the harder it is to continue increasing. That's the same idea with this pressure potential. The metric potential is the energy that's due to the cohesion and adhesion of water. All right, so the cohesion and adhesion of water refers to the water molecules sticking to other things and themselves. All right? So cohesion is water molecules sticking to themselves. Adhesion would be water molecules sticking to various structures. I didn't define those. Yeah. Didn't really define those, and I'm trying to remember if I ever do. Maybe not. So why does water stick together? Oh yeah, there it is. There's my definitions. Adhesion and cohesion. So why do they stick together? Why do water molecules stick to structures? Why do you get a water droplet that just hangs and stays stuck on a surface? Why does it have the shape that it has as a water drop? Anybody? Anybody? Why does water stick to each other? Yes?
Because it's polar.
What does that mean? Because it's polar, what does that mean?
Yeah, positive and negative charge.
All right, so it has a positive and a negative charge. Right, so what happens? Where's the positive charges?
Hydrogen.
Yep, all right, so on hydrogen. Where's the negative charge?
Oxygen.
On oxygen. Do all water molecules have this? All right, so do we have opposites attracting?
Yes.
Yep, right. So a water molecule has positive and negative. The positive is going to be attracted to the negative of another water molecule, or the negative charge of a structure, or a positive, and then the oxygen is going to be attracted to the positive charges of the structure. That's where you get this sticking. All right, so adhesion sticks to the surface, cohesion sticks to it together. Adhesion, cohesion is very important for plants because that's how our plants move water up the system against gravity. It relies on these. Now, we refer to those interactions as hydrogen bonds, all right? Those are hydrogen bonds, those forces that kind of hold them together. All right, well, the metric potential is about that cohesion and adhesion process with soil particles. All right, so the soil particles themselves can have a charge. If the soil is highly charged, it's going to be attracting those water molecules. It's going to make them hard, make the water molecules hard to move into a plant root, into a bacterial cell. All right? So water is going to be sticking to these soil particles. As water moves in the soils, water can attract other water molecules out. This is the idea of leaching the soils out of the water. You add water to your soil, the water evaporates. As the water at the surface evaporates, it pulls more waters and the accompanying solutes and salts up to the surface, where you can gradually get a highly concentrated or highly salt Salt concentrated, highly concentrated salt area right at the surface of the soil. That's all of our cohesion and adhesion. We also have to think about water sticking to plasma membranes. Is that going to be a significant amount of deal? No, not really for this lecture. We're not going to talk about that. Instead, we'll focus more on the soils, because the soils are very important for our plants. All right, so it's these three things, the osmotic potential, the pressure potential, and the metric potential that really determine how water is going to move in a system. Just to note, if we have a saturated soil, the metric potential is equal to 0, because the water that's going to be stuck is already stuck to the soil particles. All the rest is freely available to move. H bond. To go back to pressure potential. Got it? All right. All right. So now that we have an idea of water movement, let's talk about animals. So the degree of homeostasis in our animal world is going to vary. We have some animals that are osmoconformers, some that are osmoregulators. All right, so homeostasis is that physiological mechanism to maintain the internal conditions within narrow parameters, or within some set defined limits, I should say. We think of that as like temperature, but it's also related to water. All right, so we have our osmoconformers. Those are going to allow their internal water balance and solute concentration to vary with the environment. So if you go into a saltier type of area, their body will adjust and become slightly saltier. They go into like a less saltier area, their internal conditions will adjust and become less saltier. All right? So what type of organisms are osmoconformers? You can name a group, fairly large group. Sharks. I think those are regulators. You said sharks. Yeah. I'm pretty certain those are regulators. Good guess, because you are in the correct type of environment. What other organisms? Whales. Whales are regulators. Fishes are regulators. Talk about this. All right, so we kind of hit our vertebrates. What's the last group? Crabs are osmoconformers. Marine invertebrates, just in general, are osmoconformers. And you get asked, why? Why would they be osmoconformers? Because the ocean, salt concentration of the ocean is fairly constant. All right? It's fairly constant. You really have no reason to go through the expense of regulating, trying to maintain inner concentrations if you don't have to. And at least for a lot of these marine invertebrates, they've evolved earlier on. They've never really needed to evolve a mechanism to regulate their internal salt concentrations. Now you could say, well, what about some of these fishes? Fishes are a little bit different. All right, fishes, you've had secondary invasions. You've also evolved a mechanism where your body might be exposed to much more salt concentrations because the vertebrates are more mobile than our invertebrate groups. All right, fish are going to swim long, or can swim long distances, and so forth. So yeah, so our osmoregulators then are going to be those that regulate their water movement. They're going to regulate their salt balance, their water balance, and they're going to regulate it within set limits. So that individual, if we measure their osmolarity internally in a saltwater environment, if you move them to a less saltier environment, their conditions will remain the same. which that could be problematic because if you go into a freshwater area, they might not have the mechanism to continue regulating themselves, and thus they'll die. Other organisms, some organisms can. All right, so our osmoregulators, I've got our teleost fishes. That's why I said I'm going to have to check sharks because that's a different group. But our jawed fishes, bony fishes, teleosts, these are all regulators, and we'll talk about them. All right. This is a Chilean sea bass. Next time you order it on the menu, hopefully you think of this picture. Not the most appetizing, but it's good. All right, we got this? So we're going to talk about the fishes because we have both freshwater and saltwater fishes, and they're both presented with very different problems. All right, so in a freshwater system, you've got these bony fishes that live there. The bony fishes are regulating their internal concentrations where they are actually saltier than the freshwater environment in which they live. Now, because of that, the water potential is going to be higher in the fish than outside of the fish. So water will naturally want to flow out. Does it? Yeah, water's going to flow out of the fish. It's going to flow out at a fairly rapid rate. Where does it flow out? Does it flow out through the skin, through the scales? You think it does? Who's done fishing? Think it goes out through the skin? Take a guess.
It goes out through the mouth, fingers. I don't know if fish pee a lot.
They do. Well, some of them do, yeah. Do they go out? No. So so the question would be, where does osmosis take place? Does it go through the skin?
Yeah.
Where?
Yeah.
Goes through the gills. Do I have that up here? Probably. Gills are the weak spot. All right? Because what else happens at the gills? Gas exchange. All right, so the membrane's going to be thinner. Water's going to move through there, and this is where you have a lot of water movement in these freshwater fish. Actually, water's moving into that freshwater fish repeatedly. Yeah, freshwater. Water's flowing in. Did I say that? Did I reverse that? Got diagrams here and a table. So water's going to flow in. And this is a problem with all organisms, with all freshwater. You put them in there, they're saltier, water's going to move from the lake itself or the pond itself into that fish, and it moves into the gills. You get a little bit of water when they eat, all right, but that's pretty minimal. If they're going through the gills, if they didn't have a regulatory mechanism, what would happen to them? And it's the exact same thing that'll happen if you put, let's say, some marine invertebrates, let's say like crustaceans or bacteria, into distilled water. What happens to them? I'm sure some of you have done this before and not realized it. They swell and explode. They swell and explode. Our water out here is fairly salty, you can say. It's fresh water, technically, all right? But some of those organisms, if you pull out some of the bacteria, the rotifers, put them in to distilled water, you're going to see swelling. You have a complete possibility of causing them to explode. So the fish could explode unless they start to pee a lot. They have to have a mechanism to get rid of that. Now, the problem with that is that you've got a renal system that's clearing out the water. It's utilizing the process of osmosis to get water out of the tissue, out of the body, into the excretory duct, so into your intestines, ultimately, or go out through your cloaca. for these fish, all right? And they do that with the salts. So they set up a salt concentration inside their excretory system so that water will move into the GI tract and get released. All right? So out with the water is also out with the salts. So there's our problem. We're going to produce a lot of urine. It's going to be dilute, but we're going to lose a bunch of salts. So how do we get those salts back? We're going to have salt uptake at those gills. So you've got these salt pumps on the gills that will pull out any amount of water, any amount of salt that's in that water, bind to the gills. They will turn them around and move them inside. They get some salts just by consumption of their food, but they've got these salt pumps to move salts back into the body. So that's fresh water. Salt water is the opposite. Now the salt water, the marine system is saltier than the body. You're going to have water moving out at the gills, all right, which means these fish would dry up and desiccate. But the regulators, so what they start doing is they drink a lot of water. All right, you've heard the phrase, drink like a fish. All right, it's in reference to these marine teleos fishes. They just constantly drink water. The problem, though, is that along with that water is they intake a lot of salt. So I have to get rid of all this extra salt. Some of that extra salt goes out in the excretory ducts, or go out with the GI tract, all right? Most of it, though, gets pumped out at the gills. So you've got these salt pumps at both freshwater and saltwater gills, all right? In the freshwater, they pump salt into the body. In the saltwater, they pump salt out of the body, which means if you have the ability to flip these pumps one way or the other, you could be successful in both marine environment and saltwater environments. What types of fish would those be? Say what? And who? I believe bull sharks can do it. Yep. Why? Where do they live? They live both. Yeah. Do they live in like Lake Nasworthy? No. Where would they swim? Rivers. So we're talking about brackish water systems, estuarine systems. You've got fresh water coming into that ocean at that spot. That salt concentration in that area is going to fluctuate. If you have fish, salmon, swim inland, swim up rivers to reproduce, to lay their eggs, all right? They flip their pump. They spend some time near that estuaries to give themselves time to acclimate. Get those pumps, switch from pumping salts out to pumping salts in so they could survive life in freshwater environments. Here in Nasworthy, those drum can survive. They've done it. They've been adapted. They are now, or I should say acclimated. They're living in a freshwater type system. We also have naked gobies out in Nasworthy. Naked gobies are also shoreline estuarine type fish. They can live in marine environments or saltwater environments. And several years ago, we had a student that collected some and actually acclimated them so that they can live in his freshwater fish tank, or not his freshwater, his saltwater fish tank. So this is the problem. The exception for our fishes are the hagfish. Hagfish are approximately isotonic, which means they have a salt concentration, body salt concentration, approximately equal to that of the ocean. All right, who knows what a hagfish is? I know if you've had zoology, you've looked at one of these. What's a hagfish? Who's a fan of dirty jobs? Mike Rowe. Yeah, long eel-like slit fish, very, very slimy. That's the thing with dirty jobs. They went out there, they caught these hagfish, and they've got them on the table. Then they like wrap their hands around all these things, like you're making cotton candy, but it's slime that they're collecting. I mean, it's kind of gross. But what they've done, they've evolved a mechanism to actually live with that high salt concentration inside their body. They actively, maintain it so that way it minimizes water movement. Minimizes water movement. All right, so fishes, that's our freshwater versus saltwater. Any questions? They're just opposite of each other on how they work. Terrestrial animals. Oh, hold on. Let's say our quiz today has two answers. Up and down. Not left and right. Up and down. All right? Up and down. So if you want to take that, up and down. That's our answer today. Y'all connect. All right, got it? Maybe a few more minutes. A couple minutes anyways. All right. Up, down. That's our answer. Answers. All right, so terrestrial animals, their problem is drying out. Should make sense. How do they solve it? Well, you've got a couple different things to consider. One is keratinized skin, the evolution of keratinized skin. All right, keratinized skin is fairly impervious to water. Not 100% impervious, but it's fairly impervious. You can increase the amount of keratin, make it tougher, and further reduce water loss through the skin. So that's kind of like a response to the problem of low water conditions. All right, but that's more of an evolutionary response. That's not something we're just going to automatically develop. Right? It's happened over time. As our organisms have invaded land, right, you have this development, this skin, this keratinized skin. But that's not going to be enough. We also have to recover water. And we do so in the kidneys. All right, so as we kind of mentioned last week, water recovery happens in the loop of Henle. And that's what this is. All right, so water travels down, then water travels back up. And then it goes to your collecting ducts, to your bladder, and you can excrete it. All right, in this system, so we're going down through kidney. We've changed, that loop is going to pump salts out. Pump salts out to set up a salt gradient so that on its way up, water can flow out of that duct, of that loop. So minimize what we're trying to do. We're minimizing how much water actually gets to those collecting tubules. Now, the longer the loop, the better you are at recovering that water. So if we look at vertebrate organisms, and you look at those desert environments or non-desert environments, oftentimes what you'll see is those desert environments, those organisms have a much longer loop of Henle than those organisms that are not adapted to the desert-like environment. Kangaroo rats, great example. They've got a very long loop of Henle that sets up a very extreme salt gradient so they can recover a whole lot of water. We've also evolved different types of nitrogenous wastes. All right, so our aquatic organisms mostly utilize ammonia. Mostly utilize ammonia as their nitrogenous waste product. Problem is that it's highly toxic. Any of you that have kept fish know that ammonia is not our friend, not in an aquarium setting. But in a freshwater type setting, in a lake, it's not really a problem. The amount of ammonia that gets produced is small compared to the total volume of that aquatic system. So while these plants, or while these fishes are producing ammonia that's toxic to them, as soon as it gets out into the water, it gets diluted very, very quickly to harmless levels. All right? We can't really do that in our body because ammonia would cause problems internally. So what we've evolved is the production of urea. Urea is another nitrogenous waste product. It's moderately toxic. Moderately toxic. Not necessarily for us. It's not something you're going to be drinking. Pretty gross. It needs some water, though. Right? So we're going to sacrifice some water to get rid of this waste. Other organisms, such as birds, such as reptiles, produce uric acid. So this is like the ultimate waste product if you're going for water conservation. Uric acid, not very toxic at all to you. Doesn't need a whole lot of water, which means that you get rid of your nitrogenous waste, but you retain all of the water. So more desert-like organisms or those that are better adapted to drier conditions, usually produce uric acid. A lot of terrestrial organisms produce urea. And then your aquatic organisms, mostly ammonia, not a big deal because our aquatic system dilutes it very, very quickly. The last adaptation in animals is metabolic water recovery. So this is just through our metabolism. So think to, I don't know, we probably don't have many that have had biochemistry yet. This is in cell bio. You learned it in introductory bio, where you've got your sugar molecule gets broken down into carbon dioxide, water, and energy. All right? So that water can be recovered. And some organisms get by just from the food they eat. The little bit of water that's in the food, and they take that water that is an end product of metabolism, and they reclaim it. Again, kangaroo rats, great at this. Great at doing this. That's why they can live in these really dry environments with basically no water. They can recover all this water from their food so that they don't really need to drink. Will they drink? Yeah, they will. If it's available, they'll drink it. They'll drink water. Questions? All right. That is our water balance in animals. Talked about our fishes, marine, freshwater fishes. We talked about terrestrial organisms and their adaptations right here on this slide. What we're going to be moving to is our plants. All right, and the plants will take a couple lectures at least to get through some of their adaptations. All right, so there's going to be a quiz that I'm going to make go live. I think I'm going to just set up this quiz, due date sometime next week. That way you don't have to take it before our exam. This stuff is all going to be on exam 2. So what I want you to do is kind of focus on reviewing the notes for exam 1.
So where do we stop for exam?
What's that?
Where do we stop?
Right before this presentation.
Oh.
So yeah, we did thermal balance.
Writing, right?
Yep, thermal balance. Yeah, on Friday we went over Hardy Weinberg stuff. You had a question, Josh? All right. 10 minutes early. I say we're done. Not going to start. We'll start with the plans. Again, I think what I'll probably do is make this go live. It'll go live, but you'll probably have until Tuesday or Wednesday to do it. So that way it kind of gives you a break. You don't have to do it over the weekend after the exam. Gets you back into thinking about physiology. All right. See you Wednesday.